Good morning, gentlemen. Welcome to day 4 in our distance learning genetics unit, discussing the structure and function of DNA and eventually connecting it to the structure and function of viruses. So today this intro video is going to be a little bit different because I want to actually talk through a couple things. I've also been getting better with my video editing capabilities. I've been doing some screen grabs, so I want to talk you through a couple images and embed a couple other resources into this. So it'll be a little bit longer, but it will tie directly into everything you're looking at in the lesson plan today. So just to catch us up, we've been talking about the structure of DNA and how that code is read, and how it we have DNA. Hold on. How we have DNA. The instructions tell us how to build chains of molecules known as proteins, or rather a chain of molecules, smaller monomer molecules called amino acids that together form the protein. I had a really good question from Justin Samaniego about why is it that A's only bond with T's and C's only bond with G's, and then in the RNA the uracil replaces the T's and stuff. So I actually have a really good explanation for that. Some figures that I normally show to AP Bio. I'm going to do a little quick explanatory video discussing that. That's kind of its own separate piece. So if you're interested in understanding why why the code works the way that it does, that gets into a little bit more detail. Okay, so today we're going to talk a little bit more about those ultimate results of reading DNA and transcribing and translating them. These molecules, which are very important for not just the structure, but the functioning of pretty much all living organisms, especially all living eukaryotic organisms that are made up of a lot of complicated bits and pieces, because all of those complicated bits and pieces within a cellular structure are all made out of one type of molecule. Hopefully you know what it is. Proteins! So proteins are one of the larger groups of macromolecules that we've discussed, along with lipids and carbohydrates and obviously nucleic acids. But the interesting thing about proteins is that, as well as providing structure for eukaryotic cells, they also perform a lot of interesting things and tasks within this cell. Before we talk about proteins, I want to basically do a digital version of what's normally my warm-up activity for this lesson, and I want to show you a couple items here. I've got this. I've got this. I've got this, and I've got this. So if you look at all these objects, what I want you to think about is, what do they have in common? What do they have different? Think about it for a minute. This feels almost like a game of Blue's Clues, and you have to shout at the screen. Anyway, so what hopefully you've considered is the fact that all of these objects are made of metal. This one happens to have another extra coating on it because I didn't have an all-metal wrench or pliers in my garage. But these are all made of metal, certainly. But all of them perform different functions. Like this little L brace can be used to help hold up a shelf or maybe attach a bookshelf to a wall. We have this chunk of bicycle chain that's made entirely out of metal, but is very flexible and allows it to do different things. We got the screw, hold things together. Washers, used for bracing things. And then this, probably one of the more complicated pieces, allows us to grab and do stuff with that. So all these objects, made of the same material, doing different functions. And what I want you to consider is, how do we know that they do different functions? I mean, I know you know this is a screw, but what is the thing that distinguishes a screw from a washer? Because they're both made of metal. And yeah, what hopefully you've yelled at the screen, is it all boils down to shape. So when we talk about proteins, what we're really talking about are little tools within the cell. All proteins are made up of amino acids, but they're amino acids that are put into different combinations and organized in different ways. And so the way that we can tell, or basically the way that distinguishes what the protein does, comes down to how the protein is shaped. And the way that you put amino acids together changes the shape of the protein, which determines its function. Just like I could take the metal from this screw and I could boil it down and reforge it into pliers. It's still the same basic components, but the way in which all those metal bits are put together determines what it does. So when we talk about proteins, shape determines function is really the take-home message in the whole thing. But we're going to look at that in a little bit more detail. This is an image that I usually share in class on laminated handouts, but having it on screen here is good enough for the moment. What this shows are the actual protein structures of actual proteins in human and other eukaryotic cells. I know the quality of looking at this on YouTube might not be super great. I'll try to post the actual PDF of it in the folder online. But take a look at it, and I'll give you a second or two here to tell me what you notice. Okay, so what a lot of people often notice about this is, well, first off, things are really lumpy. That's cool. The colors are really pretty. Note that these proteins aren't actually these colors. This is just the way they're represented in this figure to make it easier to tell apart and to make it attractive to look at. But you see, we got some things here like these long, skinny proteins. So that looks a little bit like, you know, that screw that I showed you just there a moment ago. We got these proteins that are small and chunky and round, just like those washers that I also showed you just a minute ago. We got proteins that seem to connect things to other things. We've got proteins that have kind of weird, sort of like pinchery kind of shapes there, and all that. So yeah, what I'm hoping to have you guys kind of piece together is just like I said, that proteins are basically just like this bowl of tools on my desk are, you know, the tools of my garage. Proteins are the tools of the cell, and all these different shapes and structures help determine what it is that they do in the cell. As you look at that figure of all the proteins together, some of you might be thinking, you know, that looks kind of familiar. Like something about the colors and those lumpy shapes. I feel like this may be something we've seen before. Do you get it? Do you know where we've seen it before? I think you know. So streaming to you now, one more time, from the Howard Hughes Medical Institute, get ready to drop that shit. Okay, I'm not going to show the whole video in this video because I don't really know how YouTube censor algorithms work, even though this is an unlisted video. But I'll post the link. If you really desperately want to watch it all the way through again, I'll post the link on Schoology. And maybe I'll try doing the thing where you put the the links to other things in the thing. I don't know. Doing the YouTubes. Smash that bell, ring that button. So, yeah, just as a review, what we've talked about before and over the course of this unit, we start out with information stored in a section of nucleotides called the gene of a DNA, or the gene within DNA. And we take whatever that one section that we need and we transcribe it into a segment of mRNA, which goes through a little bit of processing before you get the final strand. Okay, great. Then that mRNA leaves the cell, where outside in the cell it is read by ribosomes and some other enzymes to translate that DNA code into a chain of amino acids, sometimes called a polypeptide, but they will eventually fold to become a protein. And just like I said, proteins are extremely important, not only in the individual cell, but in the functioning of the overall human and other eukaryotic organisms. And also, as I mentioned, shape determines function. So we have some proteins that, provide structural support, some proteins that have a bunch of like bumps and divots on them that allow them to carry things around. We have channel proteins. We've already talked about that in terms of the membrane. They're little ports that allow things in and out. And then enzymes, we talked briefly about that. Those are basically like little protein scissors that allow certain molecules to split apart or stick together. And so in order to get these functions right, you need to make sure that the shape of the protein is right. You can think about instead of thinking about a protein, think about a pair of actual scissors. If I took that actual scissors and bent one of the blades in a completely different orientation, would it still be able to cut? Probably not, or definitely not as well. So keeping these proteins within the right shape allows them to do the jobs that they need to do. So the question is, what allows them to fold into the right shapes? Well, as we mentioned before, there are 20 different major amino acids, and they can be put together in pretty much any orientation. And each of these amino acids has its own special properties. And you can see on this that they have different chemical structures. We don't really need to memorize all that right now. But you'll see that these color highlightings represent different chemical properties. So some of these are slightly acidic, some of these are slightly basic. Some of these are slightly more attracted to water. Some of these are slightly repelled from water, much like a magnet. Some of these are slightly attracted to each other or repelled from each other. So all of these things basically allow that big, long chain of individual amino acids linked together to fold up into a certain orientation. One way to illustrate this is with our friend Heimdall here. So Heimdall is a ball python, which are well known for wanting to coil up into nice little tight ball shapes. And when they do that, they want to have their heads towards the middle of the ball as much as possible. So, for example, right now he's a little bit spread out. That's pretty great. But then if I do him a boops, boop, boop. Okay, you're okay with the boops now. Boop. Okay, well, normally when I boop him on the nose, he goes, "Oh no," and does a scare and tries to coil up and have his head as deep into his little knot of coils to protect it as much as possible. So there are some amino acids that, for example, when the protein starts to fold up, they want to be in the middle of that protein. So they want to pull themselves inward. And there's other amino acids that actually want to be on the outside. And so they'll kind of pull themselves out. When I say want, I don't mean making a conscious choice. Even Heimdall is able to make, conscious choices. I mean, their chemical properties encourage them to fold in certain ways because some of them might be more attracted to water, which is called hydrophilic, or be more repelled from water, which is called hydrophobic. Normally, we do a really good hands-on activity about this that I learned from the Exploratorium, but obviously, we're going to have to make do. I do, however, have a really good video from the Exploratorium, from the Iron Science Teacher series, that does demonstrate a little bit more protein folding. It's a little bit longer, but I'm going to try just embedding it straight into this video. Hopefully the Exploratorium doesn't have content filters on it, and there's no real music to it, so it'll probably be okay. Anyway, so watch this video and keep an eye out for a special guest, not Heimdall, but somebody else you might be familiar with.
Next up, we have from the Life Science Institute. Please welcome Catherine Roquet.
All right. So I'm going to be talking about and showing you guys something that's super important, like really, really important. Like it's the reason we're all here and existing. It's so important that I think you probably already know what I'm talking about. So at the count of 3, I want you to just say what you're thinking, like what's the most important thing, like the reason we're all here and breathing and existing. So on the count of 3, just say what you think it is, all right? And I'll say it too. All right, so ready? 1, 2, 3. Protein folding, right? Protein folding. Okay. So proteins are super important. They carry the oxygen in your blood. They digest your food. Like if there's something going on in your body, a protein is there doing something to make that happen. And so since proteins are super, super small, I'm going to work with kind of a model, something that's a little bit bigger than real life. So if I could get my fellow teacher institutees to come up here and help me hold this model. So in science, right, if things are too small, you make them bigger so you can look at them. If they're too big, you make them smaller. So proteins are really, really small. So
just more volunteers. Welcome
bigger.
Volunteers. You want to let folks know your name and what you teach maybe?
My name is Nicole and I teach special ed science in Fremont.
I'm Colleen. I teach biology in San Francisco.
I'm Emily. I teach biology and anatomy and physiology at Castro Valley.
I'm Carol. I teach bio in San Ramon.
All right, sounds like a group that knows about protein
You
folding.
could just kind of like feed this down. All right. Okay, and then if everyone could take a balloon with one hand. All right, so let me take you on a little tour of this model. So what I have here is it's representing a protein strand, and protein strands are made up of individual amino acids. So something like this would be a building block for it. So I'm using tape, just the way tape is supposed to be used, right? It's just showing a connection between two things. It's bonding two things together. So where you see this blue tape, that's trying to show you that there's a really strong bond right there. There's a really strong covalent bond holding the different amino acids together. And then, of course, the other thing you notice is that we've got these balloons here, and the balloons are here to show what type of amino acid it is. So there are 20 different types, and they each kind of have special properties, which we'll be exploring in a minute. So at this point, right, we have something that we could create a fair amount of variety with, right? If you have 20 amino acids to work with, you know, we could switch the order of these, or we could make it longer or shorter, right? And we get a lot of variety. We could get a lot out of that. But proteins have a lot of different jobs. They need to do a lot of different things. And so one of the most important ways to get that variety is to change their shape. So we're going to go from a line to something that would be a little bit more useful, right? We want to broaden our toolbox, create more shapes. So I'm going to tell you guys the kind of rules for protein folding, and then I'm going to have you guys try and get your protein in that shape, all right? So if you have a pink balloon, it has a little bit of a positive charge on it. And if you have an orange balloon, it has a little bit of a negative charge. So just like you just saw in the last presentation, like charges are going to attract. So if you have an orange and a pink, you want to try and get them kind of close to each other. And then, opposite charges want to get close to each other. And then like charges, you want to kind of lean away from each other. They're going to kind of repel each other. The other options, if you have a blue balloon, it's hydrophilic. It's hydrophilic. So it wants to be sort of on the outside of the molecule because it's going to be closer to any water that's around. So blue wants to be on the outside. And that leaves us with green, that's hydrophobic, and that means it wants to kind of hide on the inside. It wants to get away from any water that's on the outside. One last rule, if you have a cysteine, so it says CYS on your balloon, cysteines really like to hang out together. So if you have a cysteine, find each other and try and be real close, all right? So those are your protein folding rules. I'm going to give you about 20 seconds. All right? So, fold. There's 2 cysteines in this strand. All right. That looks pretty good. Nice job, guys. Okay, thank you. All right, so hold it here for a second. We got to do some bonding. So, right, tape is going to be our bonding. So where are my 2 cysteines? Where are the cysteines? They're here. Okay. So I'm going to do a nice, strong covalent bond around the cysteines with my blue tape. And then do I have any positives and negatives together? Right here. Okay. So those don't form quite as strong a bond. It's a hydrogen bond. It's a little weaker. So I'm just going to kind of do like a loose tapey thing. Maybe it stays together. Maybe it doesn't, you know. Any other positives and negatives together? Okay. All right. Okay, so I'm going to try and take this and keep it shaped. Thank you so much. All right.
Thanks, volunteers.
So now we have a shape that could do something, right? This could do something. But this was only 8 amino acids long. Real proteins are a lot bigger than this. The smallest one in the human body is 44. And I clearly did not have space for 44 of these, right? So let me make a few adjustments. I want to show you something that looks a little more similar to something that a protein, shape of protein would have. So give me just a moment to make a few adjustments. All right. All right. So a few changes. So you might end up with something like this, where you get kind of twisted strands here. If you would hold on to that just for a second. And so something like this gives, here, hold on tight. Something like this could be collagen, where it gives you a really nice, strong, connective tissue, right? It's real strong. You can pull on it, and it'll retain its shape. All right, thank you. Let me give you one more example. We make a few more adjustments. All right. So something like this guy, this could be something like hemoglobin, where hemoglobin's in your red blood cells. It carries your oxygen around in your blood. And so it's got these little pockets that are perfect for oxygen. So oxygen kind of just hops on and takes a ride through your bloodstream. So this could be hemoglobin. So there's so many proteins. They all have their own special job. And so if proteins do something for you, so if you have oxygen running around in your blood right now, if you're digesting your food right now, I'd like you to take a moment and just say, thanks, proteins. All right, and thank you. Thanks.
Great, and thank you, Catherine. Channeling Michelangelo, just trying to get the cysteines to touch. All right.
What you might be thinking is, well, okay, so if gene sections of DNA tell us how to fold proteins or how to bake proteins in a certain way that allows them to fold into certain shapes, what happens when something changes in the DNA code? And that is what we refer to as a mutation. So here's a simple figure that illustrates one protein. You see, we've got some similarities here. Like we've got this, pointy triangle shape and like this curlicue here and like all these things happening on. So this is the same basic protein, but notice that they're a little bit different. So take a look at that for a second and think about what you notice. All right, so what hopefully you might have seen is that this protein here looks a little bit like, well, first we got these chloride ions are coming through. These proteins are embedded in a membrane, so they're passing through this protein out to the outside of the cell through the membrane. But this protein here looks like it's more like closed up, like that hole opening through, that nozzle might not be allowing those chloride ions to go through. Well, this specific protein that we're looking at here, this isn't just an imaginary example. This is what's known as the CFTR channel. And in a normal cell, it's open like the nozzle on a hose and allows chloride ions to move outside of the cell in like a nice little stream. But in a mutant one, there's been a change in the genetic code that causes it to form in such a way that it's tightened together. Like maybe there's some amino acids that are sticking to each other rather than repelling each other. And because of that, that's changed the shape of the protein. The chloride ions cannot pass through. And what happens is a buildup of substances on the outside of the cell that normally would rinse away from the movement of those chloride ions. Much like if your hose is blocked up, you're going to get a bunch of gunk and stuff building up on the outside. And what this is, as you might have guessed from the letters here, is the protein involved in cystic fibrosis. So cystic fibrosis, since I flipped my lecture, I wasn't able to talk about it earlier in the unit. But cystic fibrosis is a respiratory disease where people have respiratory problems in that they have frequent buildups of mucus in their lungs and sometimes also in their intestinal tract that isn't being washed away like it normally would, as a result of which people with cystic fibrosis have a lot of major respiratory problems, respiratory infections, and frequently have to go on oxygen supplementation as the disease progresses. And up until recently, cystic fibrosis was largely a death sentence by your mid to late teens. I actually went to high school with a boy who passed away when he was 16 from cystic fibrosis. And he had known his whole life that was likely what was going to happen. So it's definitely very, very tragic and frustrating. Nowadays, there are some treatments that allow people to live longer, like on average more into their 30s or so. But the real only cure for cystic fibrosis is going straight into the genetic code. So if you look at this here we have a normal nucleotide sequence, and here we have the amino acids that are coded for by each of those DNA nucleotides. And here we have what happens in the mutated sequence. And if you look at it closely, can you see what the difference is? So what is pretty subtle is there's just a couple deletions here of that C, that T, and that T, which kind of shifts everything over. So we have this nucleotide sequence ATT still results in the same amino acid, but now suddenly we skip a whole amino acid that normally would have been in this chain, and that results in an overall change in the overall protein. Another way showing it is this figure here, that this sudden deletion in that genetic sequence has a major downstream effect in the shape of the protein, how it functions, which eventually results in a change in the overall health of the organism simply because of one simple genetic change. So the shape of proteins is really important. Just like the shape of these pliers is really important to make sure that they function normally. But another interesting thing about shape determining function is that if you can figure out the shape of a protein, you can have a better sense of what it does and how it interacts with other proteins and molecules and enzymes within the cell, which is something that is actively happening right now as researchers try to understand how the COVID-19 virus is functioning and how it is entering and how it is reacting within the cells of those afflicted. Right now, researchers around the world actually are collaborating and sharing data, trying to understand the structure of the proteins on the surface of the COVID-19 virus. Many of these proteins we figured out the genetic sequence of rather rapidly. In fact, the genetic sequence of some of these proteins were determined in about 10 days, where by comparison, for SARS in 2003, the genetic sequence of the SARS protein took about 10 months to fully sequence. So vast increases in biotechnology have allowed us to respond better and more efficiently. So trying to understand the shapes of these proteins helps us understand how specifically they're attaching to cells and then being brought into cells through endocytosis. Studying the shapes of these proteins can help researchers try to develop antibodies that will stick to the proteins and allow your own body's immune system to identify these viral particles and attack them. Because frequently antibodies are a source of vaccines, which as of today apparently are already beginning trials in Washington state. So yeah, a lot of things about proteins, about why the structure of them is important, and about why understanding them is important in the context of today's modern medicine. So for your lesson plan today, if you watched through this whole video, that's a pretty good lecture introduction, but I'm also going to link to the actual slides that I went over so you can take a look at those. I will post links to some of these other videos and resources that you can check out. The main assignment is understanding the DNA code digital worksheet. That is a Schoology assignment that automatically grades you. It specifically is reviewing stuff about DNA transcription, DNA translation, and also DNA replication. So that goes all the way back. Next class, we're going to talk specifically about viruses and how they work and also how the immune system responds to them, because that will kind of connect us back together to what we've been talking about in terms of DNA and proteins, because for most viruses, that's all they are. It's just DNA and proteins. So thank you for watching. Thank you for holding in with us. Remember to wash your hands. And in fact, you know what? Just wash everything in your house, even the dog. I don't know. Might as well.